Well, good afternoon, everyone. Welcome to the first of the Bedrocan Series, a short lunch webinar with scientists from all over the world. And today is our first event. Very special welcome to Renger Witkamp.
Thank you.
Just some house rules. You can use the Q&A to ask your questions. If the questions relate to something we're talking about at that moment, I'll try to put them in, and in the end we'll end with the Q&A to see which questions are left for Renger. If you ask a question, could you please state your background to see where the question is coming from. First of all, let me start with a short poll to see who our audience is. So let me open the first poll so we can see who is visiting us before we start. Let's see where it comes. So please click on your background so we know a little bit about who is watching this webinar. Let's see the results coming in. If everyone pressed the results, let's see. We have 18% healthcare professionals. We actually have someone from the government here as well. A lot of researchers, business is mainly there, some patients and others. I don't know what the others are, but we'll see. And also, thank you very much. Next question is, because we're going to talk about cannabis as a medicine. Well, how do you see yourself? Are you an expert? Are you new to the topic? Could you please state what your knowledge on medicinal cannabis is for the next poll question? We have the results coming in, and let's see if we can get those results.
Okay. Wow.
So we have some experts in the room. Renger, that's for you. So we'll
quite a few, I would say, yeah.
But mainly about average. So we have some people actually new to medicinal cannabis. So let's see what the level is. Well, thank you very much, Renger. Welcome. Thank you for being with us. You are a professor of nutritional biology at Wageningen University, and you focus mainly on the role of nutrition, whether or not in combination with other lifestyle factors. And you have been researching medicinal cannabis since 2005 already. And recently you became the first chairman of the Dutch Institute for Medicinal Cannabis, and we would like to talk about that in the end as well. So before we come to the topic of the medicinal cannabis and cannabis as a medicine, can you please share a little bit about yourself and where your interest for medicinal cannabis comes from?
Okay, thank you, Ole. Well, good afternoon, everybody. Yes, I started as a pharmacist, so I'm a pharmacist, pharmacologist by training, but I also studied biology. So I've always been interested in bioactives, as we call them, so active components in plants and what they do to the body. So as you say, I became involved in the medicinal cannabis in around 2005. And actually in the Netherlands, these were the beginning. Well, actually people use cannabis already also in the Netherlands, but these were also the start of the start of the official medicinal cannabis program in the Netherlands. And then afterwards, I got more and more involved in both what we call phytocannabinoids, but I also became interested in the endocannabinoid system. So actually, that's what we do at the moment. So we study both the endocannabinoid system and phytocannabinoids.
Okay. And there are still many misconceptions and uncertainties about cannabis and cannabis as a medicine, both in general and in the medicinal medical world. Can you tell us something about cannabis as a medicine, the history, the functioning of the plants, and the effects of it?
Yes, sure. Well, actually, some people are really, let's say, worried about cannabis. They think it's recreational, it makes you high. Well, that's only a very small part of the story. So today we talk about medicinal cannabis, and medicinal cannabis is already it's there since thousands of years. Actually, in the old traditional Chinese medicine and also in Ayurveda from India, there has been records from the use of medicinal cannabis, and actually the old Chinese have a special character already, I think it's 2,000 years old, of cannabis. So cannabis has been used as a medicine throughout the ages. And then, I think it was until 1920 that it was still in the pharmacopoeia, so the medicine book in Europe. And, very famous people used it as a medicine. And then I think, around these years, there was a kind of opposition coming from the, let's say, the anti-addiction, like it was also alcohol. So cannabis got a kind of negative low. But, in between that, there still has been medicinal use since then. And now we are back since, let's say, 20, 25 years, in which cannabis is let's say, rediscovered as a useful medicine.
And you have been doing, of those 25 years, better part of it, you have been doing research into it. Yeah, in the beginning, it was kind of, let's say, it was pioneering. People were looking, okay, what kind of quality do we need? What kind of plants do we need? How are we going to grow? And in those days, it was also, I have to admit, in the Netherlands, we had a liberal recreational use. And I knew breeders who were, active on both sides. So they discovered medicinal cannabis, but in the meantime, they made some money from the, at those days, kind of tolerant use. So there was a kind of a strange mixture in those days, also in relation to where to use it for. So we've learned a lot during the last decades, and actually not only in the Netherlands, of course, it's a worldwide development now. Yeah. And as I said, you've been doing research for over 15 years now. Can you share your nutritional biology? So that's a very specific part of it. What is your research about? And can you share some things you found out?
Yeah, I think it was in Israel where, let's say, the fundamental work on cannabis restarted. It was in 1980 or something with Raphael Mechulam. And in 1992, his group, and in parallel a group in the UK and in the US they discovered how THC was working. And THC interacts with the receptor. And it's a kind of strange thing, of course. This receptor hasn't waited for people trying to use cannabis. Of course, it is a system which has always been there. And it's a system which does a lot of things. So this cannabinoid type 1 receptor, CB1, it was followed by a CB2 receptor that plays a major role in our body. Actually, it's one of the most abundantly expressed receptors in our brain. So it's involved in metabolism, energy metabolism, but also appetite regulation, inflammation, feeling of happiness, all these kind of things. So it's a very, very diverse system. After the receptor has been discovered, people also discovered components in our own body that interacted with the receptor. Because again, it was not, actually the plant is playing with our body, with a system which has been, let's say, developed through evolution, and is really of major importance to us. So in relation to nutrition, at a certain moment, people found out that the endogenous, as we call it, so the real ligands, so the compounds acting on the endocannabinoid system, are lipid-based, so they're made from fatty acids. So some years ago, we said, okay, well, we're not the only one, I have to say, but we were mainly looking, okay, what kind of lipids are there, and how is diet related to the types of lipids which are being formed and the endocannabinoids. And for example, we were among the first who said, okay, let's have a look at omega-3 fatty acids, which is a hot topic in nutrition. These are the healthy fatty acids. And we were among the first who said, well, okay, can we make endocannabinoids from these? Or does our body make that? And how do these work? So we are looking both from the, let's say, the medical perspective and the nutritional perspective to cannabis. And of course, there's a link with eating behavior also when you use what we call phytocannabinoids.
Yeah. And I already get a question on that in the Q&A from Frederick Lota. I can't see if it's an I or an L. He said, speaking about nutrition, because that is your thing, it's known that phytocannabinoids can enhance appetite, for instance, in cancer patients. Is that part of your research as well?
Yeah, well, I've actually not as active research, but I have been involved in a number of patients, and I've seen it. There has been studies, for example, the group of Vicky Baracos in the US, who studied cannabis, who did a trial actually, but I'm aware that a lot of people are using it for this. And what you see in many cancer patients is a symptom, a syndrome, I have to say, which is cachexia. So these people, they lose a lot of especially muscle tissue, but also fat tissue, because amongst other, they don't eat well. But it's also because of the inflammation, and cannabis has a dual action there. So it stimulates appetite. It actually gives also people more pleasure in eating. And it's not only important to the patient, but also to the let's say, the people around the patient, because they see that the patient gets more relaxed and eats better. And although I have to admit, in some cases, the situation is that bad that the patient, the prognosis is not good, that already the idea that the patient starts to eat better is very useful.
Okay. And so it can be used to enhance appetite. Can it also be used for people like me to reduce our appetite?
Yeah, well, that is an interesting thing. Actually, when the CB1 receptor was discovered, people found out that indeed, if you stimulate the receptor, then that's what THC does, but THC does a little bit more than just working on that receptor, that you stimulate appetite. And people know that anecdotally. So in those days, the pharmaceutical industry, they thought, okay, let's try to block the receptor. If you block the receptor, you might lose appetite and you may develop something which is useful in weight management. They did it. They made molecules, synthetic molecules. And when I became active in the cannabinoid field, that was around 2004-05, I said, there was a lot of interest in pharma. They were really into these, let's say, these blockers. And amongst a famous one is rimonabant. But that became a big failure because these blockers were too potent. They were called pharmacologically, they were inverse agonists. So they did exactly the opposite. So people lost indeed appetite, but they also lost their interest in many things. So that was exactly what you didn't want. So this pharmaceutical development has been terminated. However, there are some interesting compounds in the plant, like THCV, which is a natural blocker. Interestingly, in the plant, in most of the breeds, it's 50-50, THC and THCV, and that makes life a little bit complicated because the plant doesn't care what he makes. The plant doesn't make things just to cheat us. We respond to what the plant makes. But when we were able to purify or to make new strains with increased THCV and lower THC, we would be able to also to reduce appetite. But that's still in the research area, although there have been some studies on THCV that show promising results in diabetes, for example, and obesity. So there is some interesting developments there.
That's right. So if you stimulate the receptor, you increase appetite, and if you block the receptor, you decrease appetite. So actually, it would be possible. And where are you? Are you doing research on it as we speak right now?
Well, THCV we are studying in vitro still, and one of the challenges is to get it purified because THCV is rather unstable, and also it's in the plant itself. It's both there, but in the plant it has a different role. In the plant, it's part of the synthesis route. It's a kind of side product, and the plant doesn't well. It is not the idea of a plant to make an agonist and an antagonist, as you call it in pharmacology, so a stimulator and blocker in one. So we respond to THC. You can imagine if you have a strain with a lot of THCV, that the effect of THC is kind of compensated. It's a similar thing like CBD and THC, they influence each other. So that's why strain, the selection and strain stability is so important, that you have all the time the same effects.
So that is your research on nutrition. Are there any other treatments or indications you're currently working on?
Yeah, well, it's inflammation in general. We know that THC and CBD, but also CBG, we're currently looking at, is anti-inflammatory. Well, there are a lot of diseases where inflammation plays a role. So not directly related to that, it's the acute inflammation where you have, for example, an infection, but for example, inflammation related to aging, to degeneration, to recovery. Their inflammation and a low-grade inflammation, as we call it, plays a role. And what we try to do is modulate that with phytocannabinoids and the endocannabinoid group. Another area we're in is in sleep behavior, sleep quality, I would say. There are many people suffering from sleep problems, and it becomes part of a kind of vicious cycle. So we will start a project very soon in sleep quality in multiple sclerosis patients. Well, everybody who experiences a poor sleep feels poor the next day. But if the disease already affects sleep quality, then there's a real vicious cycle, negative cycle. And we try to break that by providing people with CBD in this case. So these are the main, and then we just align on gastroenterology application. So irritable bowel syndrome and inflammatory bowel disease. So these are the main things. But it's inflammation as well. Yeah.
Inflammation. So, and if any of our viewers are interested in your research, where can we find more about it? Because we only have half an hour, so there's.
Yeah. Well, we try to publish everything if we have time. So go to PubMed, go to a website. We have some, and then you can find it. So if you Google me, I think you can find some things. But I think PubMed, that's the records of scientific publication. I think the most of the audience know where to find it. Just type in my name, and I think you find some publications. The most recent one was actually one that was not given what we expected. It was on irritable bowel syndrome, CBD in a chewing gum. But it was also that we faced practical issues. So it also shows that there are also practical issues to be solved, and also in this field especially. Yeah.
So there's a lot to find on your research, and from being a renowned researcher in the Netherlands, you just became the chairman of the Dutch Institute for Medicinal Cannabis. It's a new organization, so probably many of us don't know it yet. Can you share a bit about your work and the intentions of the institute?
Yeah, well, we sat together with a number of people and said, okay, we want to take cannabis research seriously. And we also want to be a platform for education and exchange of opinions. So we decided to start a foundation in which a lot of people are represented, an organization who are, well, that's a prerequisite. We take cannabis research and application seriously. So you need to have a opioid license in the Netherlands, and you need to be part of the chain. Having said that, our network, so we are foundation, so you can't be a member, so you can, let's say, connected to the network, consists of starting from pharmacists, so people preparing real good medicinal cannabis products, to research. We have a lot of researchers. Actually, it was also for us an eye-opener that there were so many people doing research, even in a small country like the Netherlands. We were not aware of this. So that's another thing. So it's education, communication. There's also some advocacy. So we talk to the government. We want to be a neutral intermediate between users and the government, for example, as well. Yeah.
Okay. And we're at a certain phase right now in medicinal cannabis. You're doing a lot of research. How do you see the future? How do you see the future of treatments, of administration, and of products?
Yeah, well, in my opinion, there are some important points to address. Well, first of all, we need good quality cannabis. So we need reliable growers. So we need to have standard quality. We need to have stable plants. We need to have stable growing conditions. Next, I think formulations is an issue. Well, classically, it's the smoking, but we don't want people to smoke. We don't promote that. So it's about, okay, how can we administer cannabis while keeping, let's say, the good things of cannabis, which is a combination of compounds. It's the it's all the cannabinoids, but it's also the terpenoids, so other molecules, which together are very important for the action. So we look at formulation, like you should think about specific formulation for the guts that release their product locally, for example, in intestinal inflammatory bowel disease, so colitis and Crohn's disease, but also inhalation can be improved. So novel ways like powdered forms instead of just vaporizing, because vaporizing has been useful, but vaporizing it comes with disadvantages that everybody will know. So I see a lot of opportunities there. Yeah.
Okay. Well, let's move to some questions. The first one you mentioned, it's an anonymous attendee, so I don't know who's asking the question. You mentioned CBG. It's a lesser known part. Are you doing research into that as well?
Yeah, kind of in general. Well, we did some pilot studies. We know the literature that in CBG, for example, is of interest in intestinal inflammation. There have been some studies in Crohn's-like disease, but they were, as far as I know, animal models. So what we started, we have an interest in the gut-brain axis, which plays a role, for example, in when people have intestinal diseases with feelings of depression and lack of appetite. It might also might also play a role in depression, but also in loss of appetite. So what we are studying, we're looking at brain inflammation. So the hypothalamus is a part of the brain. We looked at CBG, and that's the state we're in. We look at potential anti-inflammatory effects. I have to admit, the first results are not that good. For example, CBD is a powerful anti-inflammatory. It's really great what it does. Our first studies with CBG were not that good, but I don't give up, and I think the publications at least give room for further studies. So yeah, but it's less well known. It's a minor compound, a few percent only. Yeah.
And the other one, it is. I'm not a scientist, so I hope you can get the rest of the question. Can you tell something about the difference? I think it is between cannabinoids and endocannabinoids, and how do they affect each other, the thyroid or endocrine system?
Yeah, well, sometimes to people who have never heard about endocannabinoids, I always start about endorphins. Most people have a knowledge about endorphins, but it's a kind of similar, let's say, discovery story. So first, there was the body. There was this body with all kind of systems. And apparently, there was a plant which did something. And by the action of the plant containing cannabinoids, and now I call it phytocannabinoids from plant cannabinoids, interacts with the system. And it took a while, actually it took years before they discovered how this plant was working. And then they said, okay, let's call that system, let it the same name as the plant. So the system itself works on each other, interacts, but the molecules are different. Like the endorphin was discovered thanks to morphine. Morphine is from papaver, endorphins are in your body. And the phytocannabinoids are in the plant, cannabis sativa, and the endocannabinoid system is our own system. But by coincidence, but maybe not by coincidence, because in evolution everything has a purpose, this plant, these phytocannabinoids, interact with our body, which consists of all kinds of signaling molecules, all kinds of things. And well, just for kind of arbitrary reasons, we've given that name cannabinoid system, and we call our own molecules endocannabinoids. But if you are a chemist, you would see the structures themselves are different. But apparently, by coincidence, they act on the same receptors, proteins, enzymes, and that's why they influence each other.
Okay. So actually, the endocannabinoid system was named after cannabis. Is that
it's endo? It's from your own. Yeah. But it was named, and again, that is older than the discovery of the phytocannabinoids. Okay. And animals also have it. We also studying cannabis with little worms, and they also have this endocannabinoid system. And of course, they are not designed to smoke cannabis or whatever, but they have the system, and it's a conserved system, as biologists would call it.
Yeah. I have a question. Looks like it's on your turf, from Sangeeta Natu. She says, he or she says, I'm a pharmacist and interested to know about nutritional advice to enhance the endocannabinoid system. Can exocannabinoid intake downregulate endocannabinoid system?
That's a very, very interesting question. Well, we think that if you take exo, so phytocannabinoids, that the stimulation of the CB1 receptor prevails. So that's what you actually experience. So then people eat more and they enjoy their meals more. If you look at the endocannabinoids, now it gets a little bit chemical, but one of the two major ligands, as we call it, are 2-arachidinoglycerol, so 2-HG, and anandamide. And they're made from a omega-6 polyunsaturated fatty acid. Well, in nutrition, the omega-6, omega-3 plays a role. Was a big discussion point. So what we think, but that needs to be further, well, we see that in inflammation, that by changing your diet, you get different endocannabinoids and you get anti-inflammatory effects. And with eating behavior, we are not there yet. So let's say getting people to eat more by nutrition, via the endocannabinoid, as far as I know, it has not really been successful so far. So if you want to increase appetite, I would go for phytocannabinoids at the moment in patients.
Okay. Thank you. I have another question. He said, Ryan, yes, asks for medical use or for medicinal use. Would full spectrum concentrate be advised over raw cannabis flower? So would you have the concentrate over the flower?
That's a very, very good question. It's also a question we're discussing almost every day. So you have, for those who don't know, we have the real rough plant, so the flowers, which contains everything. We have an extract, which basically contains everything, but may lose one or two components, but should still be the full spectrum, so all cannabinoids plus the terpenoids. And there's also some people say, let's take the compounds out. Let's go for pure CBD and THC. Well, there is evidence that you need the other compounds. So I would go for the combination. And then the question remains, extract full spectrum or raw preparation. Well, I tend to say, well, if the extract is good, I would go for the extract because it allows you to do more in terms of formulation. Mm-hmm. The flowers are, let's say, kind of, well, okay, they have their use, but they are kind of, let's say, old-fashioned, more difficult. You can smoke them, of course, but putting a flower let's say, in a burning device is different. It's a kind of not fully controllable way. So, as I said, I'm very much interested in specific release formulation in the gut, specific inhalation formulations, maybe dermal formulation, oral for other oral formulations. And I think then an extract, provided that it contains more compounds, would be advantageous, I would say. Yeah.
Okay, so mainly for administrative reasons.
Yeah, but also, well, the thing is, of course, you should know what's in your extract or in your flower, and people know that there's a lot of difference between them. So that makes that you should be, well, there are different preparations, as you know, with a high CBD, low CBD, etc. So standardized preparations are important, and learning to know how they work and what they do, and they differ. Yeah.
Because Ryan, in the same question, Ryan also asked, would we be able to say that all cannabis cultivars, so any cannabis out there, would have medicinal value, or? No,
certainly not. Well, I sometimes say, the most extreme case is hemp. Well, cannabis is hemp, but the industrial hemp quality, that's the same plant. But the contents of THC is very low. So people have also made all kinds of breeds. And well, of course, in the recreational field, there was a selection for high THC, and well, which resulted often in low CBD. Well, actually, there's an interest, for example, in exactly opposite. So, there will be breeds that don't do much. There will be very specific breeds that makes you very high with a lot of THC, and everything in between. So it's very important that we are able to, and we can't test them all in humans, of course. There's anecdotal evidence, so we can do inquiries among patients, but it's not that reliable because there's a lot of subjective feelings. So what we also need, actually what we do, is make kind of systems in order to screen and to compare. And we make use of fancy receptor assays. So on a chip, we make all kinds of receptors. So we screen for patterns, and we use the worms, the little worms, to see. And of course, we are not worms, but from their behavior, you can still learn a lot.
So it does matter which plant you take.
Yeah, it does matter a lot. Yeah.
Looking at the time, because we promised to do it in half an hour, but I have a lot of questions left in the Q&A. for the people wondering, it is recorded. So if you have to leave now, you'll be sent a link afterwards to see it, you can watch it back. Renger, do you have a few more minutes to answer some of the questions? Because they are.
How many questions do you have? Yeah, well, it's half an hour is finished very soon.
Half an hour is so fast. It's so fast. We want to stay, it needs to be short because people have other wanted to go on. But for the ones that have to leave now, thank you very much for attending. We'll continue a little bit with some Q&As if you're ready.
Okay. You can take a few more questions.
Yeah, we'll do some questions. And there's a question from Max Holweg. He says, due to the prevalence of cannabinoid receptors throughout the body, how often are side effects seen in the use of pharmaceutical products? I think with cannabis, I think that's the question.
Yeah.
How can those side effects be prevented?
Yeah, well, if you look at the pharmaceutical products, in the synthetic ones, the side effects were the limit. So the blockers had side effects in terms of depression, and it was indeed very difficult to disentangle the effect on the cannabinoid type 1 receptors in the periphery, so in the fat tissue, for example, and in the brain. When it comes to side effects of let's say, the herbal, so the phytocannabinoids, it's obvious, there's side effects. For example, I know cancer patients who used it, and they were high in a way. They had these central nervous system side effects. So, well, I would always say drugs which have a good action, a positive action without side effect don't exist. So well, let's say at the same time, what you know from people who use cannabis also for recreational purpose for a long time, you don't see major side effects. We've never seen cancer or whatever. So in this sense, cannabis is very safe. But again, an effect without a side effect is very difficult, indeed, given the fact that CBD receptors, CB1 receptors, are everywhere. And that's also in the body. Well, the body makes the distinguishing between the effects by changing the ligands. So there's a lot of molecules acting on that. Maybe in the plants you could do the same, but that needs a lot of, let's say, figuring out what is the balance between effects and side effects. And again, if you want to test that all in humans, that will take a lot of effort. And that's why we are in need of, let's say, model systems to do that, which are predictive. Yeah.
A little bit of advertising already on our next guest. You know him very well. It's Matthijs Pilsong, who will, in our next webinar, talk about the effects of cannabis on the brain. He's absolutely expert on that. So that's a good bridge to the next to the next webinar, actually.
You're frozen. I don't know. There is a problem. Folks, I don't know if you.
What would be the best delivery method?
So I had a hiccup in the system.
Okay.
You were frozen for a while. Maybe it's the system on my side.
No.
So could you please repeat the last thing you said about Matthijs? And then
I said something about Matthijs, who will be in our next webinar. And I said we have a question from Alex Östling. He said, for patients with pain, what would be the best delivery method? Would it be tea, oil, or capsules, or even a mix? And he says, thinking about caramelization effect versus the acids.
Yeah, well, I think of course, the bioavailability, so the uptake, the amount you take up from the gut from a tea is not very high. So if you feel comfortable, I would say if you really suffer from pain, I would try out. Because, well, basically you have the most cannabis being taken up still when you take it by inhalation. And at present, most people do it by vaporizing. So then they burn the cannabis in the system and you get decarboxylation on the spot. So as far as I know, as I see, that's still the most potent way. The oral route, so either a capsule or a tea, I would then maybe go for a capsule because it's better controllable. In tea, you never know how well it dissolves, or maybe it sticks to the cup or whatever, the glass. But still, the uptake is limited, but people who can't, let's say, evaporate, I've seen people who take it in capsules or even under the tongue. Under the tongue, actually, I forget, is also as such an interesting way. But also who took it as capsules, they have effects. But the dose is you need a higher dose because only a limited amount is being taken up. And the liver is also breaking it down to some extent.
Okay, so No clear answer.
Well, let's say the sublingual and the under the tongue. So that's why we were interested in chewing gum. So that comes there directly in your blood. So if you take it by inhalation, but I think there's room for improvement in inhalation. But then it goes directly in your blood and doesn't pass through your entire GI, gastrointestinal system, and it doesn't have to pass the liver the first time. That's what we call it first pass effect. Afterwards, blood circulation, of course, passes the liver. So there's less broken down initially. Yeah, if you take it by inhalation.
So looking at the time we're running, I thought we would be getting through the questions, but they're only getting more questions. We do.
How far are we?
When we started the Q&A sessions, there were 7. We answered a lot, and now there are 10 still open.
Okay, so let's 10 more minutes, and then we stop. Yeah, if it's allowed from the.
That's fine. I will. Okay, there's a question: how does irradiation affect the terpenes? So how does the irradiation of the product affect the terpenes?
As far as I know, there is no negative effects. There's a lot of discussion. Some people are worried about irradiation, but it's gamma irradiation. As far as I know, there are no chemical effects. It's relatively, well, it is not mild, but you wouldn't like to have it yourself, but the product itself doesn't get radioactive. And as far as I know, at the doses they use, there's no effect. I haven't seen data, clear research, but well, that's my knowledge. But you need to do it. Well, for those who don't know, you need to do it because of the biological contamination. You can imagine if you inhale cannabis, or you even despite the heating effect, some microbes, some molds, for example, could be, let's say, harmful. So that's why we need I think, a sterile preparation. Yeah.
So having a sterile product is always more important than.
Yeah, but as far as I know, the terpenes are not more or less, but also not more, let's say, changed by the gamma irradiation, as far as I know. Yeah.
And I have two or more questions on the acid cannabinoids. Someone, Stefano, says to decarboxylate or not to decarboxylate. That's the question. Yeah. What's your thought on the acid?
Thank you again for this question. Actually, more than 50 years ago, we worked, it was a PhD student, we worked on the acids. And we found out that the acids have an action. And we were, in those days, we were looking at skin, a dermal application. And we found that the acids are also anti-inflammatory. However, they don't act via the CB1 receptor. So if you want to get it into the brain, you need to decarboxylate. Actually, when you take it orally, the body also partly decarboxylates, and to make it more complicated, it adds another carboxyl group at another place in the molecule. But when you have, let's say, we still recommend for the usual applications, we recommend, well, we say you should decarboxylate. But I still think that carboxylated versions may be of interest. Yeah. Well, you can look up a publication for Kitty Verhoek's 2006 or something. It works, but not via the CB1. And we don't know yet how. Okay.
So it works, but we don't know how. Kitty Verhoek's.
Yes, but I think when you take it, let's say by smoking, you decarboxylate automatically, at least when the temperature is high enough. When you take it orally, it's partly decarboxylated. So I don't know how real it is, whether it stays decarboxylated. But in the skin, of course, that's another issue. It's stable there. Yeah.
Okay. I have a question from an anonymous attendee. What is your opinion on the efficacy and safety profile of synthetic cannabinoids? Also referring to China's recent ban on all synthetic cannabinoids.
Yeah, it depends very much what you call synthetic cannabinoids. If you have pure CBD or pure THC, I wouldn't say that it's the same molecule. So I'm trained as a chemist, a pharmacist, says the molecule is the same. So apart from the fact that you don't have the others there, so you don't have the additional cannabinoids there, and you don't have the terpenoids, I don't see any reason to believe that the action will be different. But if you talk about synthetic cannabinoids, as they are around us, then there's also a group, and that's a very dangerous group, are these fully synthetic cannabinoid-like compounds, which are misused on the street. But that's a completely different category. So don't mix them up. I think if you synthesize very well, if you do it in a safe way with no side products, then CBD can be synthesized very safely and used safely. So, but don't mix them up, because this crystalline things that are being used, people can go really crazy. You know, we have these stories of these people who were working like zombies, which was part to other synthetics, so not the same. I would like to stress this, different molecules, but based on the plant cannabinoids. So, and there were pretty dangerous ones there.
So it really depends on how you do it and what you do with. So there's not a general answer to be.
Well, it depends how you define synthetic cannabinoids.
One on your research. Are you already planning to go double-blind clinical trials, for instance, with MS patients with sleeping problems?
Actually, the MS is a different one. It's a so-called N of 1 trial, because what sometimes happened. And we saw that was our major problems in irritable bowel syndrome, that there's so much variation, both already in the disease. So irritable bowel syndrome is already a variable disease. People suffer from complaints and don't suffer from complaints for a while, etc. Everybody's different. And also the response to cannabis was different. So what we are now aiming to do is an N of 1 trial. So it's placebo controlled, it's blinded. So what we do, an on-off. So we have a treatment period, a placebo period, a treatment period, and a placebo, but we mix them up as well. So a person can have first treatment, then placebo, first placebo, then treatment. And we look all the time, we follow that patient and say, okay, what has happened? So there's fewer patients, more observation. There have been RCTs also in cannabis. And some of them were really successful, but you need the challenge to get a placebo is already a challenge, because a placebo cannabis with no effect. So what kind of plant are you taking, for example? That's another issue. Yeah.
And a question related to that, you say that it's actually, you said it, it's important for which plant you use for a specific disease, because there are different substances in it, and your body reacts differently to different substances. Is such information available about which plant is best, for example, and this example comes from Frank Glim, for example, for Crohn's disease? What cultivar, what plant should you use?
Yeah, well, I don't know about how, but let's say the cannabis pharmacy, for example, the Netherlands is now collecting these data, because the point is, of course, it will be very difficult to study that, to investigate it in an experimental way. So what you would rely on patients' experiences. But if you just send out inquiries, so then you get all kind of questions. And the question is, how reliable are they? So the cannabis pharmacy in the Netherlands, for example, is doing that together with physicians and say, okay, let's try to collect this information from patients as reliable as possible, knowing that there's always subjective effects. But that's the only way forward, I think, because there are different variations around there. Doctors prescribe different forms, and we're not there yet. But that's one of the challenges of today, is getting the right variant, the right breed to the right patient and the right indication. Yeah.
So, well, we're really running out of time now. We went 15 minutes over time. We still have some questions left. We will answer them in writing. So I'll send them we'll send them to you. Make sure they get to you, Renger, so you can answer them. And someone also asked, I don't know what is already in the chat, but via the email that you registered with to this webinar, you'll get the link so you can watch it back and you can watch all the answers. Renger, thank you very much. Is there anything, a last word you'd like to share with us or the participants?
Well, it has been a pleasure. And I think, well, thank you for the questions, people. And yeah, it has been a pleasure. It's a really interesting topic.
Absolutely. So, you know a lot about it, and you showed it. So if there are any questions for Renger, don't hesitate to ask them to us. We'll make sure they get to you. They can also contact you directly, probably. Your research can be found on PubMed. Thank you very much. We also put the link for the Institute of Medicinal Cannabis in the chat, so people can get more info on that one. And I'd say thank you very much. It was the first in the series. I hope you enjoyed. If you did, please register for the next one. The next one will be, as I said, with Matthijs Bossong on the effects of cannabis on the brain. And I wish you a very pleasant day. I don't know in which country you are, if it's the same temperature as in the Netherlands, so it's really hot here these days. Thank you very much, and thank you, Renger.
Okay, thank you. Bye-bye.
Bye-bye.